Most of us learn a very simple equation in middle school. Plants take in carbon dioxide and water, use sunlight, and produce glucose and oxygen. It is often treated as basic biological plumbing. But if you bypass the surface of the leaf, drop into a single cell, and push into the chloroplast, you arrive at the thylakoid membrane. This membrane is packed with a highly specific pigment, chlorophyll A. At this scale, converting light into physical matter is governed by the laws of quantum mechanics. To figure out exactly what dictates a plant's ability to grow in the macroscopic world, we have to reverse engineer this biological engine from its subatomic foundations. The thylakoid membrane is built like a microscopic sandwich, consisting of alternating lipid and protein layers. Suspended exactly within these layers is chlorophyll A. The molecule has two distinct parts: a hydrophilic magnesium porphyrin head that rests snugly inside the protein layer, and a lipophilic phytyl tail anchored firmly down in the lipid layer. That tail acts as an anchor, holding the molecule perfectly steady, so the porphyrin ring at the top can do the actual work of absorbing incoming radiation. But running this machinery comes with a severe hazard. The process constantly interacts with highly reactive oxygen molecules, which can easily oxidize and destroy the delicate chlorophyll structures. To survive this, the plant surrounds its chlorophyll with accessory pigments, like carotenoids. These absorb some light energy, but they also act as a physical shield, intercepting those reactive oxygen molecules before they can cause photodynamic damage to the system. These pigments represent a highly organized, physically hardened solar antenna array designed to capture light while surviving chemical fallout. The entire mechanism triggers the exact moment a single photon of sunlight strikes the magnesium atom inside that porphyrin ring. The photon's energy forces an electron out of its normal ground state, kicking it up into a higher, highly unstable energy level known as the excited second singlet state, before it rapidly drops into a first singlet state. This excited state has a lifespan of roughly 1/1000th of a second. If the plant fails to harvest that energy within that fraction of a second, the electron falls back to its ground state. The absorbed energy is wasted, radiating away as a red fluorescent glow, or simply dissipating as heat. To prevent that total loss, the antenna array performs a Forster resonance transfer. Instead of dropping back down to the ground state, the excitation energy hops laterally, resonating from one pigment molecule directly to the next. This resonance effect rapidly funnels the energy inward, bouncing it through the antenna array until it strikes specific reaction centers, known as P680 and P700, which can actually use that trapped charge to drive photochemistry. Photosynthesis is a biological race against time. The plant must capture and pass along this fleeting energy before it decays to a ground state. Because this system relies on the strict structural geometry of these specific molecules, the antenna array cannot process every type of solar radiation. It only works with precise wavelengths of visible light. This chart, plotting the absorption spectra of isolated chloroplast pigments, maps out exactly how much light these molecules capture across different wavelengths. You can clearly see massive spikes where absorption peaks. These occur almost entirely in the violet-blue range at one end and the red range at the other. When we look down at this middle chart, the action spectrum, we are measuring the actual rate of photosynthesis in a living plant by tracking its oxygen release. The curve of biological activity perfectly mirrors the chemical absorption peaks plotted directly above it. We can see this proven physically in this bottom diagram of Engelmann's historical experiment. By shining a spectrum of light on a filament of algae and introducing aerobic bacteria, the resulting layout is clear. The oxygen-hungry bacteria aggressively congregate in the exact blue and red bands where those pigment molecules are active. They clustered there because that is where oxygen was being pumped out, proving the biological engine operates strictly on those blue and red wavelengths. By combining the subatomic properties of these isolated pigments with physical biological output, we can map exactly how a plant processes the electromagnetic spectrum. Yet, for all the subatomic activity happening inside the thylakoid membrane, the plant as a whole still has to survive in a highly variable physical environment. In perfect conditions, the theoretical efficiency of this conversion is roughly 70%. It takes exactly 4 photons of light to transfer the 4 hydrogen atoms required to reduce carbon dioxide into usable carbohydrates. But in reality, perfect conditions almost rarely exist. the plant's overall output is tightly governed by the principle of limiting factors. This first graph tracks the rate of photosynthesis against light intensity. At point B, a lack of light restricts the rate. Moving to point C, the line flattens. The quantum antenna array is at full capacity. Additional photons won't force the process any faster. We see a similar constraint in this second graph, measuring carbon dioxide concentration. At point D, the rate hits another hard plateau. Even with infinite light energy, the chemical factory reaches a maximum speed limit and cannot process carbon molecules any faster. The final bottleneck is tracked on this temperature graph. Initially, the rate climbs steadily as heat provides kinetic energy, speeding up reactions to the peak at point A. But past that peak, the rate catastrophically crashes. High heat denatures the proteins, melting down the apparatus. Photosynthesis functions as a masterclass in biological engineering, an engine driven by quantum transitions and bounded by the rigid limits of environmental availability.